raw. The problem is the conditions at which sprouts are grown are similar to the conditions at which bacteria thrive. Outbreaks of sprouts infected with E. coli and salmonella happen fairly regularly. In 2010, 44 people from 11 states got sick. Sprouts are one of the few foods that many people grow themselves in their homes. FDA scientists have discovered the cause of most contamination starts with contaminated seeds. Therefore, even if sprouts are grown under perfectly sanitary conditions, harmful bacteria from the seeds can flourish. The focus of our project is to see if the consumer can reduce or eliminate bacteria present on sprouts by soaking them in salt water. We met with a grocer and vegetable expert named Venus Perez at Whole Foods. She thought our idea might work, but she warned us that sprouts are very fragile.
Let's take a look at an alfalfa sprout's food journey. The food journey starts with the seed. The seeds are harvested from an alfalfa plant using a combine. Then the seeds are separated from the plant with processing equipment, dried, packaged, and then shipped to seed distributors, either for home sprouting or mass production sprouting facility. In both the home and commercial sprouting, the seeds are sprouted for about 4 days in a humid and warm environment.
Unfortunately, there are many places of possible contamination in this journey. When alfalfa plants are grown to produce seeds for sprouting, possible sources of contamination include infected soil, water, and animal waste. During harvesting and packaging, the seeds could be contaminated by dirty processing equipment, ill employees, pests, or unsanitary packaging materials. During commercial sprouting and distribution, unsanitary conditions and improper storage temperatures could cause sprouts to become infected.
We did our own lab experiment to test our hypothesis that soaking contaminated sprouts in salt water will make them safe to eat. All sprouts have plenty of bacteria on them. They are only dangerous if they have been contaminated along their way with harmful bacteria. We wanted to see if our solution worked on both benign and harmful bacteria. So we purposely contaminated a batch of sprouts with raw chicken that had been left out for a day. The lab experiment we did consisted of soaking contaminated and uncontaminated sprouts in various concentrations of salt for 10 minutes to see if we could reduce or eliminate bacteria. According to our mentor, pathologist Dr. Joe, high concentrations of salt might inhibit but not kill bacteria, so it would be important to test both unrinsed and rinsed conditions. We then cultured bacteria on a nutrient agar-filled Petri dish from each of the 5 salt conditions, rinsed and unrinsed, and contaminated and not. We designed and programmed a LEGO NXT-controlled incubator to keep the bacteria at 35 to 37 degrees Celsius, the ideal temperature for bacterial growth.
In designing our experiment, we searched to see if studies have tested salt solutions as a disinfectant and found only one study. The study showed that a 10% salt solution wasn't effective in eliminating E. coli, so we tested up to 15%. Our research did not find any successful methods for treating sprouts for bacteria. The only treatments we found were involving seeds prior to the sprouting, and these were not 100% effective. Sprout production companies periodically run lab tests on the sprouting wastewater to make sure that their products are safe.
After incubating our petri dishes for 24 hours, we analyzed our results. The fact that our negative controls grew no colonies tells us our method of taking samples was good and our supplies were sterile. The uncontaminated results showed that as salt concentration increased, the number of bacteria colonies decreased for both rinsed and unrinsed sprouts. The rinsed samples didn't, in general, show more colonies, so it seems that the increased salt concentration was actually killing bacteria and not just inhibiting its growth. You may notice an anomaly in the uncontaminated 15% category. We attribute this to a few mix-ups in procedures. The results of sprouts contaminated with raw chicken are hard to interpret because we had a lot of trouble pouring the agar for these. The agar had clumped up, and many of the features seen in the petri dishes are agar clumps and not bacteria colonies. The 15% unrinsed did show some clear colonies, so it may be that harmful bacteria are harder to kill than benign bacteria. We would need to retest to be sure.
We found 10 documents on the FDA website addressing the contamination problems of sprouts. Currently, the FDA does not recommend any procedure that can be done in the consumer's house to make sprouts safer to eat. We presented our results to the grocer Venus and our pathologist mentor, Dr. Joe. We taped our presentation and put it on YouTube as well.
One discouraging result we found was that the grocer Venus was right. Sprouts are very fragile, and soaking them for just 10 minutes in 15% salt solution makes them mushy and unappealing. This opens the door to new inquiry. Perhaps longer soaking time and lower salt concentration could make the sprouts safer to eat without making them mushy. We think we had a good idea, but just chose a particularly fragile vegetable to experiment on. Our basic idea is easy and cheap to implement and may prove beneficial for killing bacteria on fruits and vegetables with tougher skins.
So is this just a linear increase, or can you describe the correlation as with the salt increase and the decrease in bacteria?
For the uncontaminated, it worked really well, a salt solution. As we increase the amount of salt, the less bacteria grew.
So, like, you can see there's like colonies right there. Do you see the colonies? And then here, 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 and there's less as you progress. If you compare that to that, and there's a lot less.
These aren't colonies, these are agar clumps, and these are colonies, so the colonies are sort of yellowish.
Yeah.
So easier to see when they weren't photographs, but we couldn't bring them actually in.
Yeah. And then for our contaminated sprouts, it did the same thing. The more salt concentrations, the less bacteria, except for 15%.
Yeah.
That one had surprisingly a lot of bacteria.
Yeah, but we had a mistake when we were trying to test it because we accidentally rinsed the sprouts before we tested them. Like, we were supposed to have unrinsed and rinsed, and we accidentally rinsed them.
So you rinse them after, or with the contaminated ones, you rinse them before you put them in with the chicken? Or, I was a little confused as to what's the difference between rinsed and non-rinsed is.
Okay, so rinse. So the reason why we had to rinse our sprouts were because, well, we tried the salt concentrations, and then the reason why we had to rinse is our mentor, pathologist Dr. Joe, she said that sometimes the salt can inhibit bacteria growth, so she said it'd be a good thing to rinse the sprouts to make sure to get all the salt off so that we could also get another reading.
Like, so that the salt didn't get into the actual petri dishes, it just killed the bacteria on the sprouts.
Gotcha. As far as the bacteria, do you know if they were good bacteria or bad bacteria, or just the fact that it was bacteria because it grew in the petri dish?
We have no idea.
Okay.
We didn't research that.
But we left chicken out for a day, so that should collect some harmful bacteria.
Hopefully. That's why we did the contaminated.
How'd you come up with the idea?
Well,
we spent a long time brainstorming. We actually didn't start our project until like a third of the way through the season. So we were back and forth between a lot of different things, and then we thought, well, alfalfa sprouts are very easily contaminated, so if we could figure out a way to fix that, then that would be great. But it turned out that it doesn't work as well for alfalfa sprouts because if you see that little picture over there, they got mushy.
Yeah, they get mushy
so
and unappealing.
we're hoping that it would work better on some other fruit where it wouldn't get all mushy.
When you were talking about when you shared some of these ideas amongst each other and with others, what kinds of comments or feedback did you get?
Well, we shared our results with our pathologist mentor, Dr. Joe, and she actually told us that we did the experiment a little bit wrong because we should have had a control for the rinsed also.
Yeah.
So she's basically saying that all our rinsed are not good, are bad because we didn't sterilize the water completely. So we didn't test that the water was sterile.
Was it just tap water that you guys rinsed with?
Well, it was distilled water.
Okay.
Yeah, but we didn't test it for like if it was sterile or not.
Gotcha.
And so we also shared with Venus, and when we were sharing with her, we were at Whole Foods, and some people actually stopped by and started watching us throughout the presentation. So they didn't send any feedback because they sort of just left once we were finished.
And you looked at other solutions and compared, or how did you think of the salt thing?
Well, like
we were going to try and test with lots of stuff like vinegar and xylitol, different types of vinegar, different concentrations, except for we didn't have enough time and enough petri dishes and stuff like that. But the experiment that we found with the E. coli, someone tried to kill.
But it was just E. coli and not other forms of bacteria. So we decided to just try like harmful bacteria in general, which is the chicken left out for a day.
That was the person who tested only up to 10 and convinced us to go to 15.
And other studies were for decontaminating the seeds, not the sprouts. It was just like applying chemicals to the seeds, but we're testing the sprouts, like decontaminating the sprouts.
Well, your time's up, unfortunately. Yeah, 10 minutes goes by real quick. So.